Here's a clear essay-style explanation of the differences between Archaea and bacteria. The difference between Archaea and bacteria. Archaea and bacteria are two of the three major domains of life, the third being Eukaria. At first glance, Archaea and bacteria seem very similar. Both are single-celled microorganisms, lack a nucleus, and reproduce asexually. For many years, Archaea were even classified as a type of bacteria. However, advances in molecular biology, especially in the study of ribosomal RNA, have shown that Archaea and bacteria are fundamentally distinct groups. Their differences span genetics, cell structure, metabolism, and ecological roles. 1. Genetic and evolutionary differences. Archaea and bacteria differ significantly at the genetic level. Archaea share certain genetic features with eukaryotes, such as similarities in the machinery used for DNA replication, transcription, and translation. In fact, archaeal RNA polymerase and ribosomal proteins are more closely related to those of eukaryotes than to bacteria. This suggests that Archaea and eukaryotes share a closer evolutionary relationship, even though Archaea physically resemble bacteria. 2. Cell membrane composition. One of the most striking differences between Archaea and bacteria lies in their cell membrane structure. Bacterial membranes are made of fatty acids attached to glycerol with ester bonds. In contrast, archaeal membranes are built from isoprene chains attached to glycerol with ether bonds. Ether bonds are chemically more stable than ester bonds, allowing Archaea to survive in extreme environments such as hot springs, salty lakes, or highly acidic conditions. Some Archaea even have monolayer membranes, which further enhance their stability in harsh settings. 3. Cell wall differences. Both Archaea and bacteria typically have cell walls, but their composition differs. Most bacteria have cell walls made of peptidoglycan, a polymer unique to them. Archaea, however, lack peptidoglycan. Instead, they may have cell walls made of pseudopeptidoglycan, proteins, or polysaccharides. This distinction is important because many antibiotics such as penicillin target peptidoglycan synthesis, making bacteria vulnerable but leaving Archaea unaffected. Metabolism and environmental adaptations. Bacteria and Archaea also differ in their metabolic capabilities. While both groups exhibit great diversity, Archaea are especially known for thriving in extreme environments, earning them the title of extremophiles. Some Archaea produce methane as a byproduct of metabolism, methanogens, a process not found in bacteria. Bacteria, on the other hand, are more common in everyday environments and play central roles in processes such as nitrogen fixation, decomposition, and human health. 5. Pathogenicity. A crucial difference is that many bacteria can cause disease in humans, animals, and plants, while no known Archaea are pathogenic. This makes bacteria highly relevant to medicine and public health, whereas Archaea are more often studied for their ecological roles and unique biochemistry. Conclusion. Although Archaea and bacteria are both prokaryotic and superficially similar, they are fundamentally different at the molecular and biochemical level. Archaea are genetically closer to eukaryotes, have unique membrane lipids, and thrive in extreme environments, while bacteria are abundant in more common habitats and include many pathogenic species. Understanding these differences not only clarifies the tree of life, but also highlights the remarkable diversity of life forms that exist on Earth.